Okay, sorry I can't be at the Bridge project dinner. I had a trip planned a long time ago, so the new date didn't correspond to it. I'm going to be talking about diagnostics in my laboratory now, and in particular this project on MR readable sensors. Basic idea behind this project is that we are coupling, as many projects in my lab, we're coupling existing medical procedures with something else, like a diagnostic in this case, or a drug delivery, or something like that. The idea is we can accelerate some of these technologies into the clinic by making them compatible with existing medical procedures, medical practice. The prototype for this project is the medical biopsy. It's sort of the gold standard for cancer diagnosis and staging. There's many different ways to get a piece of tissue. The one illustrated in this slide is this needle biopsy shown here. It's, of course, while it's probably the most important cancer diagnostic, it is also can be quite invasive, and so it's hard to use this very important diagnostic through the course of therapy. And so what we're doing is trying to tweak this a little bit, use that existing procedure to deploy a small piece of material that can be interrogated non-invasively many times after deployment. I'll talk about a couple of them in this talk, it's a 20-30 minute talk. The first is based on MRI contrast agents that are inside the device. We've worked for a few years, 4 or 5 years now, on this particular concept. The idea behind this contrast agent is that it's functionalized so that it can change its aggregation state in the presence of particular analytes. And we can detect the state of aggregation of these iron oxide particles with an MR scanner. Briefly, the background on this is an early version of the devices that I'll talk about. This was for subcutaneous deployment in animals. It consists of a couple of precision molded plastic pieces, a functionalized sensing agent that's described here, that's isolated inside of the device by a semi-permeable membrane. So these small iron oxide particles can't get out through the membrane, but the analyte that we're interested in can enter. And this just shows you what one of these things really looks like. The first study that we did with these devices functionalized with antibodies is to look at how the signal measured by MR varies with concentration. And what we found was that it didn't vary with concentration. It actually varies with exposure. Antibodies on the surface of these particles functionalized for a particular analyte have a very high affinity. And as a result, there's sort of, once they've aggregated, they really don't come apart once you lower the concentration of analyte. Instead, how these sensors work is they aggregate, and the amount of aggregation depends on how long the analyte has been present. If you take the analyte away, no more aggregation occurs. And so what you see in this plot here are 3 different concentration profiles in time. So in this case, you get 50 nanograms per mil for so many hours. It goes down to 0 and so forth. And here it's even still more complicated. exposure profiles. And what you find is that all of these exposure profiles give you the same curve on the right. That's because T2 versus what I'm plotting here is total exposure, which is the integral underneath these. So this is a very interesting kind of sensor. When this sensor with these high avidity particles exposed to an analyte, it's like a dosimeter. It's measuring the exposure of the device to the thing you're interested in. And this has prompted a whole new avenue of research in my lab, because we're now looking at ways of measuring very, very small concentrations of analytes by using this kind of an approach, where we're trying to pull the signal, a very small signal, out of the background by basically integrating the exposure. This gives you an idea of what these kind of devices look like in the scanner. This is sort of a false color image, but basically what we do is measure the magnetic properties of the hydrogens within the device. And we can basically do a T1 or T2 measurement at this, in this small volume using the scanner and the Koch. This was an early study where, just summarizes an early study where we gave mice myocardial infarctions and looked for proteins look for proteins associated with an MI. So troponin, myoglobin, CK, and these were compared with animals where we did a sham procedure, and also those animals where we didn't do anything to them. And what you can see in all three cases, sensors for troponin, myoglobin, and CK identified those animals that had a MI. So that gives you an idea, or this slide gives you an idea of what you can do with these high avidity, basically dosimeter-like sensors. What I'm now going to switch to is a sensor like what you're more used to using. That is a sensor that's reversible, that is maybe doesn't have quite the specificity that an antibody does, but is nonetheless has the advantage of having a measurement that's correlated with concentration. And the one I'll talk about is hypoxia. In this part of the project, what we're doing is making materials that are sensitive to the local concentration of oxygen. So the thing about this is, rather than injecting a contrast agent and having it go everywhere, we can go back to the same location that we left this material and measure over the course of time the degree of hypoxia. And in cancer, as you know, that basically the tumor structure or tumor metabolism is such and histology is such is that there are regions within the tumor that are very hypoxic. And this hypoxia is already known to be clinically relevant for things like in this slide, what it's describing is the use of radiation dose compared with how hypoxic the tumor is. And what the finding was here is the more hypoxic the tumor, the greater the radiation dose necessary to get the same effect. Now, of course, you've probably heard of other ways to do in vivo oxygen detection. Indeed, there are MR methods for doing this. The disadvantage of these that depend on contrast agents, the advantage of doing it this way is, of course, it's non-invasive. You can inject the contrast agent and get what you want in the MR. It does provide the spatial resolution that you wouldn't have with a device located in a single or multiple devices in selected locations. But the disadvantage is that it does require the contrast agent. It's susceptible to the distribution of that contrast agent, which isn't known. And it's one of the reasons why this type of technique has never been used and probably will never be used for making absolute oxygen content measurements. What we've done is to switch this around and look at common materials used in biomedical applications and ask, can they be used as oxygen tension hosts in the MRI? So the example I showed here are silicone type materials, analogs to things that you're very familiar with, PDMS. Medical grade silicone is this material that you see here on the lower right. It turns out that these silicones are very hydrophobic. They have a very high solubility for oxygen. Oxygen's paramagnetic, and as a paramagnetic solute in the polymer, that paramagnetic solute will affect the relaxivity of protons on the polymer. So if I take a piece of silicone and deploy it in a piece of tissue, it will be saturated with oxygen surrounding it, and the effect of the concentration of that oxygen will depend on the local oxygen concentration in the tissue. That will then affect the relaxivity of the protons in the silicone, and we can measure that with the MR. At least that was the hypothesis. Here's an example of a specimen polymer. This is a 1 millimeter hatch here that is small enough to be injected into the animal. And there's such an image in the animal. You can see a very small device image in the animal, and we can do the inversion recovery sequence just on that material. So that's we'll put the region of interest around here, capture this data. Okay, so here's the last couple of slides. This is response to oxygen in vivo. So what's happened here is I'm comparing the image intensity in a region of interest separate from the device to a region of interest that contains basically the device. And we switch the inspired gas from air to 100% oxygen. You can see over the course of 20 minutes, change in intensity of the device, no change in intensity in the control region. And I know it's hard to see in this image here, but what you see here is one such device. We, in an inspired gas study in animals, this is one sequence, an inversion recovery sequence. This is what happens to the image intensity as a function of time. And again, you can see a T1 type curve, or relaxation curve is here, and we just measured the time constant from here to get an absolute T1. You see over here. Here are 4 different animals where we've done the same sort of experiment that I've described before. So this is oxygen, air, and oxygen again in this sequence. And so what you see is that we do indeed get repeatable reversibility in the animal as we go to the high oxygen content. So now the animal experiments are geared towards doing hypoxia. We have an approved protocol, and what we're doing is we deploy these devices in the limb of a rat. We have a cuff that can controllably restrict blood flow to the limb, and we are now driving that limb hypoxic with application of pressure in the cuff. So that pretty much gives you a broad overview of what I'm doing in my diagnostics part of my lab. There's more to come, but if you have any interest, drop me an email. I'm at mjcima@mit.edu. Thanks.